So, without thanking them, I don't think I can celebrate my achievements. Celebrating my achievement is celebrating my PhD students. So, three of them, Sarah, Carla, and Kevin, already graduated. Kevin's viva was last December, and they are in various places. Kira is finishing up her thesis, so soon to be a doctor, hopefully. And Sam, 2 more years with me, I think. So, whatever I'm going to say in later in terms of my research is our contributions as teamwork of different people involved with my research. Then I'd like to thank a lot of people. I mean, this is the pictures I can find that fits within one slide. So it is not an exclusive set of pictures, but I have a lot of friends, colleagues within Swansea, and beyond, collaborators, mentors, which played a vital role in my career so far. So having that important bit done, so I can go to research. So I thought I'll start with my time in Singapore, right? So Singapore, I went there to do PhD. So I was doing a project in a discrete convolution theory based on a professor who was in Singapore. So I wanted to work with him. So when I went to Singapore, he moved to US. Maybe someone told him or about me. So Professor Ping Limping kindly took me as his PhD student. Then I started working on heat and mass transfer in bread baking. It's collaboration with food technology department. So after a few months, I thought it's not something I wanted to do forever. So that's the time I saw a lecture given by Siv in Field Institute of Mathematical Sciences on how maths can be used in biomedical applications. So I thought I reached out to Siv. Then he said, fine, just apply. That's how I went. I finished with the master's there. Then I went to Waterloo. So the funny thing, Professor Ping, I again saw him in Dundee. So when I went to Dundee, he was a colleague in Dundee as the chair of numerical analysis. So the world is too small. So then I started my PhD in Waterloo. So that's a bit snapshot of my PhD thesis. So I was looking into mathematical modeling of gliomas. Glioblastoma is one of the aggressive cancers. So we are trying to look mathematical modeling of gliomas and treatments, especially radiation therapy, and the factors that affects the efficacy of treatments and so on. So in particular, hypoxia. Hypoxia is the state where cells lack oxygen as they grow. It has hypoxia, and oxygen is one of the important prognostic marker in treatment responses. For example, in radiation, you need oxygen to fix the damage caused by the radiation. So you need that oxygen for the treatments to be active. So we're looking at different problems related to that. Just to mention two key research from that PhD studies is, so we developed a mathematical model for studying radiation therapy, the effect of radiation therapy in combination with chemotherapy. And it's one of the early models in that area. And we also looked into how, so hypoxia, as I mentioned, hypoxia is an important factor in treatments. So most of the times in clinics, in reality, what we do is, what they do is, not me, they take biopsies and other little things to know whether the tumor is hypoxic or not. But sometimes beyond that, you like to know what is the spatial distribution of the hypoxia, where the hypoxic regions are. So I'll come to a lot of, hypoxia is a common theme that I will be talking in throughout the talk, throughout this talk, because that, oxygen considers one of the main factors that driving many things in cancer. So what we did is we tried to compare image analysis with modeling to see, having known the image of vascular structure, the spatial distribution of the vasculature, can you predict hypoxia? So that, for example, there are imaging techniques like angiogram. It's not refined enough to get all the minute details, but having known something, some information about the vessels, can you predict what's the hypoxia and oxygen distribution and so on? So that's the main two themes that I focused on my PhD. So we went to interstitial fluid pressure and all those stuff. But yes. So now I think this is a good time to pause and think, why are we doing this? Right? What's the point of doing this modeling? So to me, modeling is all about finding, now the keyword is digital twin, right? So to have a way of giving individualized treatment to an individual patient. So right now what we have is a uniform solution to an individual problem, which means if 10 patients come with a certain cancer, most of the times these 10 patients get similar treatments. But they may have different backgrounds, different behaviors or biological scenarios and so on. So can we use the information that you get from MRI scan, biopsies, blood work, all those things, put into the mathematical model and say, okay, for this patient, this is the best treatment strategy. We are already doing that for weather. Weather is a complex problem. We are predict. We know the weather was going to rain next in an hour or two. So can we do the same thing with cancer modeling? Okay, I'm not there yet quite, but that's the way I like to take this. And we can also use mathematical modeling to test clinical trials, experimental designs. I will give you a few examples as we go along. Predict optimal drug dosing and sequencing. Most of the times, the cancer is treated with the multiple therapies, chemotherapy, radiation, immunotherapy. What is the best way to combine them? Should I give chemotherapy first, radiation second, or the other way around, or to give it together? Things changes the way you give these therapies to the patients. So we cannot test everything. We can. It's very costly experimentally, but it's always good to have a lab in your computer to test in a cost-effective manner, so that we can select things that you ideally want to take into experiment to test these things. Okay, so in a nutshell, we're trying to simulate a little lab. I know a lot of people here are modelers, and they know what I'm talking about. So trying to simulate a little lab in your computer so that you can do experiments in that little model. So in PhD, I was looking into a big, large-scale behavior of cancer cells, how cancer as a concentration evolves over time, how it responds to therapies. But sometimes you like to know a little more details from that. Like you like to know, what's happening within the cancer cells. So cancer is a complex disease with a lot of processes happening within the cell, outside the cell, how it evolves, interacts with the environment it grows. For example, if you plant a tree in a fertile soil, it grows really nicely. If you, plant it in a different place, it may not. So it also matters where the cancer grows, the microenvironment where things grows. So like oxygen distributions, other factors play a key role in this. Then how everything evolves over time, because all these processes happens in different time scales. Some are in seconds, minutes, some are in hours. For example, how a cell grows, 24 hours, 16 hours, depending on the cell line. So to put everything in a proper, meaningful way, we try to define a multi-scale approach. Again, probably this is the only slide that a lot of maths in there. So we try to model what's happening within the cell by set of, for example, a set of ordinary differential equations, or it can be modeled different ways. So we're trying to say, we're trying to incorporate things that's of our interest, intracellular level, extracellular level, such as oxygen distribution, which can be modeled by using a partial differential equations, and then put them all together using a multi-scale techniques in a meaningful manner. So we were looking at individual cell-based approaches, such as cellular automata, cellular ports models, or there is a physical framework that we recently started using it. Then put all these informations together to get a multi-scale framework with which we can play. So just two movies just to show what is the implementation of this mathematical model. So the first model, so here what I'm doing is I am planting a tumor cell within a normal tissue, for example. I'm assuming the normal cells only grow if there is a free space, which means outside. and the cancer cells grow uncontrollably and see what's happening. So you can really see how cancer evolves in a normal tissue. Different colors represent cells in different phases. For example, cells go through cell cycle phases, G0, G1, etc. So sometimes it's good to know those phases, because some of the chemotherapy drugs that we give are particularly effective in certain phases of the cell cycle, including radiation therapy. Radiation therapy is active during the phase where DNA divides. So it's good to know these phases just to get an idea what kind of treatments we need to give. The next one is you can model a 3D tumor as well. So we assume there is a lot of pipes of vasculature. Then oxygen is coming out from these pipes. Then you plant the tumor cells there. Then how it grows over time. Again, these are the models that we create so that we can use this. We can use this to test different other things later. So this is a simulation created by Sarah during her PhD. So these colors represent the concentration of oxygen. If they are nearby the vessels, they have more oxygen, away, less oxygen. So it's good to know this spatial distribution of oxygen for therapy purposes. So then you can look at how it compared to experiments. It's always good to produce good videos and nice little pictures, but does that make any sense? Okay, so we can try to compare our simulation results to what's really observed biologically. For example, the inside part here is hypoxic regions where they lack oxygen. Outside you have cells that grows. Okay, you can get similar kind of behavior in silico models as well, and you can study how it evolves over time as well. Okay, so as I mentioned, you can create a model, you can get a nice animations, you can get nice funny pictures, but to make sense of that, you need to tune your model. You need to calibrate your model with experimental data. For example, different cancers or different cancer cell lines behave differently. They grow with different lengths, and they respond to the treatment differently. So it's good to include those details into the model, to tune the model first. So we did this work with Institute of Cancer Research London. So Sarah was a PhD student at ICR London at that point. Now she is a team lead in ETH Zurich for mathematical modeling. So she did the experiments on different cell lines. Then based on the experiment, we selected key parameters, key points that we need to feed into the mathematical model so that it can fit the data. So it's like you are tuning, you are making your model or calibrating the model for that particular cell lines. So once you tune your model for that particular cell line, then you can ask questions like how this cell line responds to different treatments. So in this study, what we did is we tried to analyze, we tried to find the effects of the radiation combined with heat given by high intensity focused ultrasound, HIFU. So HIFU is heat is a radiosensitizer, which means it will help the cells to respond well to radiation. So what happens if I combine this heat with radiation? So that's the experimental combined modeling study that we did with Sarah. So once you have this calibrated, tuned model, you can study many things. You can study theoretical studies. For example, drug resistance is a key factor that impeding chemotherapy response. Right? For example, if a patient is given chemotherapy consistently, same drug, same dose for a long time, they don't respond very well to the chemotherapy after, so drug resistance. So we try to analyze what are the factors that contribute to this drug resistance. There are intracellular heterogeneities in terms of cell cycle changes, different pathways, and external factors such as oxygen distribution, the spatial distribution of oxygen, how it evolves over time. So all these factors kind of feed into this drug resistance in cancer. So we can analyze all these things by using a little lab in your computer, all right? And this image shows G1. G1 means if I give a drug that kills the cells in the G1 phase, and another dose of the same drug, what's going to happen? So we're trying to find out if I give you 2 types of chemotherapy, what is the best way to combine them properly? So now we can also use mathematical models to find something or study something unknown as well. So this is one of the studies that I want to give as an example, where you can use mathematical models to provide hypotheses to test after. Okay, so we were studying the role of radiation bystander effects in after giving the radiation. So the bystander effect is, so when the cells are radiated, the cells die or they repair or they have after effects of radiation, right? So now sometimes when cells are radiated, so these red cells, the cell that is nearby, which is blue, which is not radiated, behave like they are radiated. That's a bystander effect. It's like somebody is attacked in the next door, we will run away or do something else. So that's the bystander response. So the cells behaves like that. So there are experiments that can, that can quantify this. It's really hard to quantify it. So some experiments are on, like you in a petri dish, you radiated one cell, then try to see what's happening in the nearby cells. Or there are experiments like medium transfer experiments where you transfer the medium and culture new cells, then see the effects. So long story short, so what we wanted to study is the effect of this bystander effects in radiation. So if you look at this curve, so this is a dose response curve. So the x-axis you have dose levels, and y-axis you have surviving fraction, means how many cells are survived after that many doses. Higher the dose, more cells are killed. Lower the dose, less cells are killed, right? But if you look at that part, you can see there is a higher cell kill when you have a lower dosage. Okay, so we call it radio hypersensitivity at low doses. So there are different explanations. One explanation is at low doses, the cell's repair mechanisms are not really active. They are not awake yet, which means more cells are killed. But what we propose that radiation bystander effects may also play a role in to have that low dose sensitivity, to have high cell kill at the low doses. We used our multi-scale mathematical model, including all the biological information, biological observations, and to study these radiation bystander effects. And we also found, that if you simulate the mathematical models, you can see the red curve shows the effect of cells if I target directly the cells, and the blue one is with bystander effects. And you can see at low doses there's a little dip initially, which kind of show there's a higher cell kill initially. So this is later a Carmel mother cell from McMaster. They showed experimentally that at low doses there is a role played by bystander effects in inducing the low dose radiosensitivity, and they even cited our work in their paper as well. So this is one way of, you do theoretical studies and how it motivates experimental findings later on, and rather than the other way around, right? It's a two-way scenarios. So which then led to different interaction with Carmel's group and beyond. So we were working with a group under International Union of Radioecology on radioecology. So this is a group of people from different countries. So this is one of the posters that we had during a NATO sponsored conference in Yerevan, Armenia, on environmental effect of radiation. What happens if you have accidents like Fukushima? What's happening to the environment? So there is ecological point of view as well as is there a role for modeling in that as well. So we call these papers as a duck paper, badger paper, and bunny paper in a nice way. Okay, so that's where, how the models, how the interactions can promote interdisciplinary collaborations in a wider scale as well. So we still keep interacting together. So the COVID kind of stopped everything in between. So we are going to restart everything again. So now in next few slides, I'm going to show or summarize the research that we have been doing in past few years. So as an examples, what modeling can do, what mathematical multi-scale modeling approach can help in terms of cancer treatments. So this is on hypoxia-activated prodrug. I will explain how this works. So this is one of the drug they introduced, saying this may be the next big thing in cancer treatment. So how it works is, it's a nice cartoon made by Philip Group. So as I mentioned, the hypoxic area, so here they don't respond very well to treatments, whether it's a radiation therapy, chemotherapy, okay, I'm playing with that, okay, or different treatment, it won't respond very well. The idea is you give the HAP, or hypoxia-activated prodrug, it will go to the hypoxic area, and they are activated by hypoxia, lack of oxygen, which releases the drug, then it kills the cells that are hypoxic, then other treatment can be more effective. Okay? So as I mentioned, okay, so the next slide will show you what is the hypoxic area. This is an experimental figure where all these highlighted areas are hypoxic area, and this is after one dose of this HAP drug, CP506. You can clearly see, hypoxic cells are killed. So once it's killed, there is reoxygenation. You can give other treatments. Yeah. But preclinically, in experiments, it's very highly successful treatment. But clinically, it was not showing too much success. So most of the clinical trials are not very effective. So we were reached out by a group in Netherlands, asking whether we could analyze it mathematically, or can you develop a mathematical model to test or find scenarios or find biomarkers that can be used to select patients, for example. What are the key factors playing a role in this effectiveness? So this is a work done by Sarah doing her PhD here. So we used the multi-scale model with all the equations and all the maths behind it. So we can use experimental data. For example, this first set of figures show the experimental data where a tumor grows. All these green cells are cells that are hypoxic. And similarly here, all these green cells, they are proliferating cells. They have enough oxygen. So we can tune our model. So this is a way of, you have a model, first you tune your model for that particular data, particular problem. Then you can develop a mathematical model, including all these experimental observations. You can implement that, find that model so that you can work with that after. You can ask questions, what if? What if I switch that off? What if I change something? So it's a little tool for you to play with. So that's what we did. We tried different scenarios in discussion with our experimental collaborators. So there are two things that I'm just highlighting here. So for example, these two tumors have same hypoxic fraction, means same amount of hypoxia. But one is there is a big core of hypoxic cells. The other one is you have like islands of hypoxia. But in tumor, there is no guarantee that you have a big core of hypoxia cell. It's very fragmented sometimes. So we saw that in the study, from the study, what we observed is it's not just the hypoxic fraction, but it's the spatio-temporal evolution or distribution of the oxygen or hypoxia that really matters to know the effects of this HAP. So which means to select the patients, we like to know the not just hypoxic fraction, but how this hypoxia is distributed spatially. Otherwise, sometimes you won't notice the effect of HAP, or HAP with radiation, or HAP alone, or radiation alone. You won't notice the difference. So to notice the difference, you need to select patients so that they are, this treatment is effective. So the next work is from Kira. So this is in collaboration with AstraZeneca. So we are trying to model something called DNA damage response inhibiting drug, or DNA damage response pathway inhibiting drug, DDR inhibitor. So when cell grows, so this is a cell cycle. If you know your biology of behind, so cells go through and they have to go through a process before it divides. So when they go through this process, there are a lot of checkpoints, just to see everything is all right. If not, I won't let you pass. You go back, get prepared yourself, and come back. So they have to go through this checkpoints to have a successful division. But that's where the DNA damage response pathway is responsible. If there is a damage, these pathways repair it before it can proceed. Okay? So the idea is we are going to give drugs that blocks this repair. For example, if you have cancer cells, you target cancer cells with radiation, which means you create damage, induce damage to the cells, then you don't want the damage to get repaired. So you give the drug that stops the damage so that you have higher cell kill. The treatment that you gave is highly effective. There are a lot of drugs at the moment. Olaparib, I think it's clinically available now. Patients are given those drugs. I can't pronounce Selarcitinib. We call AST some numbers, which I think is also available. So we're trying to study the combination of these two drugs and in combination of those drugs with other treatments such as radiation therapy as well. So why we use this? Because in normal cells, there are other repair mechanisms that are active. Even if you target certain things, it's fine, they will be okay. But in cancer, everything is kind of complex, and a lot of things are not working properly, so it may be able to target them efficiently. So we received the data from AstraZeneca, and based on this paper, and then what Kira developed is a compartment model, a set of ordinary differential equations based on the key interactions that play a vital role there. Right? This is in discussion with experts in AstraZeneca to incorporate everything into the model. Then we have a mathematical modeling approach. We have a mathematical model. Then we use the data that provided by them to tune these parameters for that particular cell line and data set. Then we used a different set of data to see whether are we getting the right things or not, validate the model. Right? Then what we want to do is we want to combine that with radiation to provide predictions. What's going to happen? What if scenarios? What's going to happen if I give different doses of the drugs and so on, so that possibly that can be taken to experiments for trying. So right now we are trying to incorporate this is a ordinary differential equation based modeling approach. We are trying to incorporate that into a multi-scale approach, so that we like to know what is the effect of spatial changes, right? Whether the spatial changes incorporate or induce drug resistance within the cancer cell. So the next work, again, everything in terms of cancer theme on understanding the effect of environment, for example, oxygen, on developing different phenotypes in cancer, right? So here, Kevin used a data analysis approach where there are a lot of data available online, mRNA data, gene expression data. So we try to analyze the data using pathway enrichment analysis. We try to identify the key pathways which are deregulated in cancer. Then we try to incorporate that into mathematical models to see the role of those in eventually in treatments. So again, we adapt our mathematical model to include those details. Then we can analyze how cells behave, how their cells evolves in different environment, different scenarios, and make predictions based on that. Or what is the role of oxygen? What is the role of oxygen? Drives a lot of pathways such as HIF, hypoxia inducible factor pathways. HIF has a major role in regulating or deregulating many other things. So how it drives different metabolic changes within cancer. And we also looked into, so while we do this data analysis, we found that one of the pathways that is deregulated is collagen pathway, which is essential part of extracellular matrix. So cancers grows in an environment. For cancers to move around or invade other tissue, it has to degrade the matrix. So collagen is a big part of this matrix. So again, in this case, we try to adapt the model to see what is the interaction or, between collagen and cancer and other microenvironment and so on. Again, we can analyze different scenarios. For example, this is everything is fine. So here you have a hard matrix, and one part is soft matrix, and how things evolves accordingly. What happens if you have heterogeneous distribution of oxygen, or everything as a mishmash complex matrix, how things changes, which probably the reality. Okay, and finally, almost there. So Sam is working with Professor Darina Gonzalez from medical school. So we are trying to analyze the role of adipocytes or adipose tissue in cancer evolution or cancer aggressive nature of cancer. So they are working on ovarian cancer. So in omentum, there are a lot of adipocytes. So depending on the type of adipocytes that you have in omentum, you can see some of the cancer cells are aggressive in nature, which means they don't respond very well to the treatment, and they are quite different depending on the adipose tissue concentration. So we are looking in particular two cell lines called SCO and OCAR. They behave differently. So the inside part is kind of red, you can't really see. So red kind of shows the cells that are mesenchymal in nature, which are aggressive. They can invade other tissues. And the green cells are kind of epithelial marker, which means they're all right. But there is epithelial to mesenchymal transition is a key factor of cancer evolution. So going from epithelial to mesenchymal means cancer is becoming more aggressive. So we're trying to develop a mathematical model now with a physical framework to analyze what are the key factors playing in this transition, why these two cell lines behave differently, and what are the factors driving this evolution and so on. So again, this is a different slices of this spheroid. It's a 3D dimensional tumor. So we're trying to now tune the mathematical model by using the experimental data that we have. So to analyze how it responds to treatments, chemotherapy, and why one of the cell line behave differently and so on. That's where we are. Okay. With that, I'm going to summarize. I think I'm right on time. What I kind of showed here. So as I mentioned, the main key motivation and aim of my research is to, can you create that digital twin, right? Can you create the model that can help with treatments? Can I, if a patient comes in, if I know that data such as MRIs, and if you have MRI data at two different times, you can try to guess what is the growth rate of a tumor in a way. But the structures, where it grows also matters. For example, in brain tumor, they grow differently in different tissues. So gray and white matter, the brain tumor grows, the growth speed is different. So we need to analyze that other factors into account as well. So having known all these things, can you give a recommendation saying, oh, for this patient, this is the best treatment outcome or best treatment strategy? It's easy to do in simulations because we are not harming anybody. So we can create possible scenarios that may be effective, that can be taken for trial, clinical trials and so on. Experimental trial designs, I gave an example how a theoretical study can motivate experimental studies. To see things that you can't see experimentally, for example, if I switch off, what will I get at the end? Can it similar to what they observe experimentally, so that you can identify the key players, in that driving certain things, etc. So I'd like to thank many people. I mean, these are a short list, which relevant to the materials that I presented. And there are, people from mathematics department and FSE. It's a maths department is a small little family where everybody supports each other. It's a nice little environment to do your research. And finally, I'd like to thank my family. And without their support, nothing is possible. Things like staying late sometimes, working on weekends. So especially Tina and Daniel, they are in the back. So thank you very much. And my family, probably they are watching from home. And I also take this opportunity to wish happy birthday to my dad. Today is his birthday, so that he may be watching there. Thank you. And with that, I'll stop.